Hello, Science 30s. Hopefully this is our last little video on what to do with our biology. And this is going to be the last little bit on our biology talking about inheritance and how do we actually follow these traits that get passed along. Now that we've talked about genes and we understood what's happening with the DNA, we're going to actually look a little bit more in depth and we're going to start talking about these traits that get passed along. And so these traits are acquired or they're inherited. You can acquire traits like scars, or you can inherit things like your hair color and different things like that. So, two more terms. We're going to have a few more terms and then we're going to put some practice into the video near the end as well. So I'm going to go over some terms and hopefully we're practicing and following along with our workbooks to help make sense of all these new terms we're learning. So what is responsible for all these differences we have in these various traits that we have? We're going to call them alleles. And these are an alternate form of a specific gene or genotype. And so tongue rolling, you have to have two alleles for this trait. Individuals who possess these two alleles for this trait, they might be able to roll their tongue, or they might have a certain type of hairline, whether it's straight across or it kind of comes to a peak. And with alleles, we're going to learn about that there's a dominant allele and a recessive allele. And so you kind of have these two types. You can either show this dominant trait or you have this recessive trait. And when you have two recessive traits, because two sets of DNA, right? We're having a mix up of it. When you have these two recessives, you show the recessive gene. So with these alleles, or we're going to talk about this phenotype, and that's going to be the actual way we're going to look at it. And we're going to use letters, and we're going to use capital letters and lowercase letters to describe the phenotype, whether it's dominant or it's recessive. So I talked about hairlines, and you can see number 2, the hairline. We have this thing called a widow's peak, and right here, the dominant one would be two capital W's or a capital W and a lowercase w. Both have the dominant gene, meaning the person would show this widow's peak, having it kind of pointed. However, if both parents were able to pass along the recessive type gene, because remember when we talked about genes and passing it along, each parent passes along half of their own genetic material. So if they each get that half that's both recessive, they have this straight hairline. Like I said, hair type could be curly or straight. Free floating ear, so whether it's attached or it's free floating, or left or right handed is even the traits that get passed along. And so if you're right handed, chances are you have this type of gene or this phenotype. If you're left handed, you might have this. One thing I'm going to warn though, it's not just one type of gene or it's just one thing that determines what we look like and how do we look like, but we're going to simplify it and use it as a very simplistic look to start with. So like I said, you're getting half the material from your parents and you only need one dominant allele for the trait to be expressed. You need both recessive alleles to actually show the recessive trait. Pretty straightforward. So we have this autosomal inheritance, and we heard that word when we talked about mitosis and these autosomal cells. And so this is all the 22 pairs. So the 23rd was the sex cell. So these are the other 22, and these are called autosomal inheritance. And we can have chromosomal disorders that can be used to diagnose through amniocentesis, and we learned about that in our last video lesson. But all of these genes, where we're talking, let's say hair or our earlobes, these are autosomal inheritance. They are physical features. On the sex chromosome, so on that 23rd pair, we have the X and Y, and these are the ones that determine gender. And so we differentiate between sex but not other phenotypes. And so these genes, anything linked to these 23rd ones, are called sex-linked inheritance because they're on those sex chromosomes. And so something like color blindness, where you can't differentiate the differences between red and green, or sometimes people just see shades of gray, these are passed along on the X and Y genes. So before we jump into figuring out how do we determine what our phenotypes might be and what alleles might be passed along, we want to talk about this guy called Gregor Mendel. I just want to give a quick little summary about him. He's thought of as the father of genetics because he ran experiments on pea plants. It's an organism, and they pass along information from their father to their son, or their what we are going to call the parent to the daughter. And so he started to record, okay, we have these two parents. What kind of traits do they pass along? And he only looked at a select few. So he looked at flower color, or the shape of the pea pod that was formed, or the actual roundness or the wrinkliness of the peas themselves. And he had about 9 of them that he followed and tracked. And he decided when he was looking at them, sometimes these traits would get passed along and this dominant trait would be showing up all the time. However, sometimes the recessive traits would come back and he would see a pop-up of this recessive trait. And he tried to figure out how do genes get passed along and why do some show up more often, making them more dominant, and why do some kind of just skip a generation? You don't see them in the one generation, but then you see them in a second generation after that. So he was the first one that started with all the recording, and he started the process of the thought of, oh, we pass along information from parent to the offspring generation. When he started to notice things, and he was recording it over hundreds and hundreds of pea plants and years of data, he started to notice that it started to fall into percentages. And he started to notice that it was either like 100%, 75, 50, 25%. And so it was kind of like this broken up into quarters. And he used a table that he could determine the alleles of the parents to show possible outcomes for this fertilization. You can't say 100% that, yes, this plant will be this one. Well, it's like flipping a coin. You have a 50-50 chance. Well, with these Punnett squares, it's a 1 in 25% chance. So when we talk about them, we're going to use these capital letters to denote our dominant alleles. We're going to use lowercase letters to show our recessive ones. And then we're going to have a table, and I'm going to show you on the next one, on how to actually solve these. And so we're going to have examples like a capital T or lowercase t right here, where it's dominant, but it's split between a dominant and a recessive. We can have two dominant ones like this, or we can have two small recessive ones. And so the first two are going to be, actually, I'll do it in blue, make it a little more clear. These first two are going to be, oh, the plant is going to be tall. The second one, if they get two recessives, the plant is going to be short. And so we're going to have terms for these. Like I said, we're going to have some terms that we're going to need to learn through as we're practicing. So make sure we're getting and practicing with these terms and using them when we can. Two terms we're going to use are homozygous and heterozygous. So homozygous meaning both the alleles are the same. They can be both capital or they can be both lowercase. And so we might say something like, oh, homozygous dominant or homozygous recessive. But then we know what that is. Heterozygous tells you right away, one capital, one lowercase. We're going to use this term, it's a carrier. It shows the dominant trait, but they are carrying a recessive and they can pass along that recessive gene. So here we go, right from our textbook on like page 99, it was an example of how we draw out a Punnett square. This is great, but for the first time we're learning how to do a Punnett square. I'm going to walk you through on the next slide on how do we actually plan it out and figure things out. So we're going to use a sex-linked example because it's a little bit more detailed, but yeah, it's pretty much the same when we talk about autosomal as well. So this is how I like to draw it out. I draw a 3 by 3 kind of block, but I leave the tops open and it's just quick, easy, 3 lines down, 3 lines across, and we connect them, boom. And so what we take from the question, if you went back to it, it was you had a mother who was homozygous full color vision, meaning she was all one type. So a mother is XX, all females are XX, and we use this capital N, capital N. So we can see it's sex-linked, so we're going to put in the sex gene, but then we're looking at the two dominants being capital letters. And I'm just using no blindness. We are using N. The father was colorblind. And so the thing with sex-linked is it's carried on that X chromosome. That Y doesn't have any notice to it. Okay? And so if they have the Y, that's not the determiner for this gene. It's all based off of that X chromosome. So what we'll see now is we need to place it in the table. So what we're going to do for the mother, we're going to take one of her alleles, put it there, take the next one, put it there. Same thing for the father. We're going to put one here on our table. Now you can see, okay, the mother and the father, if they were to pass along one of their traits, we put it in the table. Same thing. If we choose this one, the father will put it in and he'll pass it along. So what will happen is this child will be female because it's XX. The female will also have full color vision because it still has the dominant allele there. It has that capital N, meaning full color vision. Only an X with a lowercase n and another X with a lowercase n would mean color blindness. Or in a boy, if it was XY and they had the X with the lowercase n, like the father, they would have color blindness. And so as we fill out the rest of the table, as we bring it down, we fill it out like this. And so what we see, and if we went back to the question, it walks you through the answers. Both of the chances that you'll have a girl, right here, 50-50 chance. Both of the chances that you'll have a female, both will be not colorblind, or they'll have full color vision. Same thing for here, because the mother passes along always this X1, that's homozygous. They always have an X capital N, X capital N. Both of the boys potentially will have full color vision as well, because the Y does nothing for this color blindness. So essentially, the females can become that carrier for it that we see right here. They can carry and pass along that gene right here, but they will not present or they will not show it. Both of the boys will not get color blindness either. So before you go on to blood, please make sure that you are practicing some of these Punnett squares. They're pretty straightforward, but understand the statistics of them and practice drawing them out and practice questions with Punnett squares. Because now that we're getting into blood, we're going to twist it up a little bit, and it's not always just as straight simple as dominant recessive. When we talk about blood, we can see there's things like A blood or B blood. But then what happens is there's an AB blood, and then there's an O type. And so blood is what we call codominant as an example. Sometimes this happens as well where there is no just recessive, it's a codominance. Both of the traits present themselves. And so you can see the genotypes for this A blood. You can have two potential types from your parents. A parent could be AA and pass along only the genotype A, or a parent could be AO, meaning they could pass along the marker for an A-type blood or an O-type. And so it makes it a little bit more complex when we're talking about blood typing. It's just not as simple as, oh, 4 blood types. There's a lot more in depth that we're starting to know about blood typing. And so this is a good example of codominance. So like I said, practice with these Punnett squares, practice with the percentages like, oh, do you have a 100% chance of inheriting this trait or passing it along to your offspring? Is there a 50% chance? Is there a 25% chance? Remember, it's not guaranteeing that, oh, you have 4 children and they'll always be all dominant. No, it's a statistics. Flip a coin twice and the chances that it's both heads, well, we have our chances, right? Chances that it's both tails, there's those chances, right? Same thing with genetics. It's statistics. And so with these statistics, we're also going to get interesting things called mutations. And so what happens with these mutations is something goes wrong with our genes. And so we talked about that, and now we're coming full circle back to chemistry where we talked about UV radiation and benzene rings. And this is actually a mutagen, where it's an agent or it's an outside source that causes mutations to increase. And it's something that's there to cause a mutation, like cancer. A carcinogen or a cancer-causing agent increases your likelihood of having cancer. So smoking is a carcinogen. The things in there cause your cells to mutate to form into cancerous cells. And so are my example a little less dire than smoking is all horrible, but X-rays and radioactive decay, they also are carcinogenic and they cause cell mutation leading to cancerous cells. So with these mutations now, this is in the person and the DNA replication actually has a mistake made or there's changes into the sequencing. And so we talked about how these base pairs match up in DNA and these mutations is when the cells make a mistake. And we're going to have 2 different types of mutations on this cellular level. And so we have a point mutation and a frame shift mutation we're going to look at. I pulled this one right from our textbook, so it's good to be familiar with some of the ones they outline in the textbook, just what exactly they are. And we're going to see that like cystic fibrosis or Huntington's disease and hemophilia, we're going to focus in on a couple of these and we're going to see what exactly is happening. And now with our technology, where we're actually able to gene map, we're actually able to say, oh, chromosome 7 is responsible for this type of mutation or this type of disease. Or chromosome 4 is where you can locate Huntington disease and you can find these markers of, oh, your parent or grandparent maybe had Huntington disease. You can have a DNA test and they can look at chromosome 4 and say, oh, do you actually carry the genetic markers for Huntington disease or not? So, talking about point mutations and frame shift mutations. This point mutation is where the amino acid is altered. And so remember when we had this coding with the CTCGGA and how it copies? Well, maybe there's just a single letter that doesn't copy quite right. And so sometimes this can be for the best, or it can be for the worst, or it can actually just do nothing. And the amino acid's also altered, but maybe it's just the same one and it still makes sense. So our example is our 3-letter runs, like we have for amino acids, the fox can run. Instead, the box can run. Kind of doesn't quite make sense, but it's still readable. Maybe the amino acids are still the same. Like when we saw that table, we had 4 codings for some amino acids, so maybe it still codes for the exact same protein. A frame shift one, on the other hand, is something completely different. And so you can see the fox can run is still there. However, it's like we've taken the letters and we've shifted the whole run of 3, and we've added an A at the front of the fox can run. And here you can still see it kind of weird to look at the start, but there was our starting 3, our next 3, next 3, and then there. That's where it was, but everything was shifted and now it makes absolutely no sense. And so this could be completely and totally different, making it not a desirable mutation at all. So Most times when you hear mutations, either you think of the X-Men or you think of something very, very bad. And so some mutations are actually good and it helps animals and just everything survive because they are able to pass along traits that are maybe a little bit more desirable, giving their offspring a better chance to survive as well. The example is here shown is the peppered moth. Initially, the trees were all white and this moth was able to blend in. During the Industrial Revolution, all the trees turned black and these flies or these moths had to go through a rapid mutation, a rapid change to re-blend in so they just didn't get picked off by all the birds. It's very easy to see the yellow and the white one on a black tree, but when the trees all turned black because of pollution during the Industrial Revolution, they had to go through a quick mutation and rapid change so they could re-blend in with their environment. And so it wasn't they could decide to be black, it was, oh, all of a sudden the ones that were a little bit darker survived, passed along those traits. Those traits got passed along and along and along until the moths mutated to a new color. Here we're going to lead into now is pedigree charts, and I want to talk about hemophilia and the good and bad about it. If you don't know what hemophilia is, this is where you're able, or you're not able rather, to actually have your blood to cause a blood clot, or when you get a bruise, you keep bleeding. When you get a cut, you don't have any of these platelets to actually cause your blood to clot, and you don't have the ability to stop bleeding. So it's very, very dangerous. The reason we look at these pedigree charts is from way back when we look at royalty, and people would always want to know how they're related or how they are royal, and so they always kept long lineages of family trees. But here we see how hemophilia gets passed along through family trees. And you can see it starts right at the very top. Was a carrier female, meaning this Albert, because he had a son right here, somewhere along the line. Oh, okay, he shows this hemophilia. And then there's another carrier that comes in and then marries in. The issues are when these two lines, they come together. And the family, or royal families, unfortunately, start this inbreeding. Or even more recently, as we look around here, these are very close cousins. So it was a brother or two sisters at the top here, and here, two sisters, and then the cousins got married, and you can see all of a sudden, now they pass along these traits, and hemophilia just doesn't work its way out. It actually becomes more and more prevalent because of inbreeding within these royal families. So that's the dangerous part about these mutations is depending how genes get passed along, you can have this dangerous, dangerous effects. And so there's lots and lots of examples of different royal families. So here you can see the inbreeding of Charles II of Spain, and you can actually search up the Habsburg chin, and that's what you see right here in the pictures is the chins that stick way out, and so it's a mutation, but it was a dead giveaway of you were part of this royal family. You passed along this trait, this Habsburg chin. So this one's a little bit reversed because it's at the bottom and it's going up. But you can see this uncle and this niece were married, first cousins were married, and it's highlighting how first cousins and second cousins were marrying, or uncles and nieces were marrying and passing along too similar of genetic material, passing along this chin feature that kept on through generations. So let's actually jump into pedigree charts. How do we actually read it on a very much more simple case? Now that we've kind of seen some very complex ones, we're going to read it on the very simplistic sake, and we're going to practice with some of them. So they consist of symbols, and they identify known genotypes, so unknowns can be predicted. We have this in our data booklet, this pedigree chart, and it's going to tell you all the little details that we have, whether it's shaded in, and those are affected individuals, if they're shaded. You have squares to identify a male, you have circles to identify a female, and then a line between them on the same generation line means they are mating together. And so we see the generations here. There's one, this Roman numeral separates, there's generation, so we could think of it as grandparents. You could think of it as parents, and then we could think of it as kids right here in this third generation. And you can go and backtrack, okay, they're affected, meaning there must be a recessive somewhere, and these recessives can be traced back. And so we're going to practice some questions and practice in your workbooks how to solve pedigree charts. So first one here we see this autosomal recessive. So remember these autosomal traits, and they're passed along on those 22 chromosomes. And so this autosomal recessive means there's a recessive trait that's showing up, and the shading is a recessive genotype. And so what we see here, we could say, oh, cystic fibrosis. So let's just go R, recessive. And this mom, we're not sure. Well, we could take a guess though, because 1, 2, 3, 4 kids that they had, none of them showed. I could take a guess it might be R, R. 2 capitals, 2 dominants. It's not likely she has it. The one thing I do know down here on generation 3 and child 4, they would have 2 lowercase r's, meaning an R would have to be here and an R would have to be here. Since they don't show the trait, they also have the dominant one. Okay, and this is where we need to practice our pedigree and our Punnett squares together, because we're trying to plan out and figure out the statistics of what allele do they actually pass along. So hopefully you practice Punnett squares and we understood how to pass along these alleles, and we can predict and work backwards almost as the easiest way of how do we understand the pedigree chart. Also with pedigrees, we also have autosomal dominant. And so this is where the disease, like we have here, is Huntington's disease, is a dominant one. And they pass along a dominant trait. And so you'll start to see them pop up every generation. The last one we looked at, it missed that generation, then another one popped up from the previous generation. So it might have just been carriers from the second generation, but then you see it pop up in the third one. The dominant one is you're starting to see it every generation because, well, it's a dominant trait being passed along. So it's going to pop up more and more frequently. So when we're looking at genotypes, like I said, first one, it's going to tell you if it's autosomal recessive, autosomal dominant, or if it's going to be sex-linked. But first things first, autosomal recessive. So right away, I'm just going to choose a letter. Easy one for me to draw is an R line, and it's easy to tell which ones are capitals and lower cases. I can fill in all the recessive traits. And this is my clue. It's kind of like a big puzzle that we can go through, and we can go backwards to figure them all out. Now, even though I said this is autosomal recessive and it's popping up lots, we have to be careful because we need to see what's what. And so I'm going to look on this side right here, over here to start, and kind of work my way back to solve it. So when I look at number 14, I don't quite know right yet. So I'm going to go up to number 6 right here. And for number 6 to have an offspring that is recessive, that means they must carry something right here. They must carry a recessive gene to pass along a recessive gene, so their child has recessive. Meaning their other one, because they're not showing the trait, is dominant. Okay, and so working back up all the way to number 2, well, did they get the recessive gene from their mother or did they get it from their father? Well, if their mother had a recessive, they could, I don't know. We'll keep problem solving. And so as we work through these, it might take you some practice with some Punnett squares to figure out what are the chances? Or with number 2, can we figure it out? Well, let's work backwards again and work with our recessives and try to backtrack again and figure out and try to problem solve and take educated guesses. When we look at number 3, let's say, for them to have number 9, number 10, and number 12, oh, they have a child that has double recessive, meaning this parent must have it right here. They must have a recessive gene to pass on. So they must be a capital R because they're showing the dominant trait, and a lowercase r because they do have a recessive child. Meaning the other children are, well, let's do a Punnett square quick. Like I said, I can do it up to this way. And the father is lowercase r. Here is capital R, lowercase r. We need lots and lots of practice. Like I said, we should be able to do them quite quickly, and you'll get practice in your head. And so there's a 50-50 chance that a child will have RR, or lowercase r. Child 9 and 12, there is no chance that they have two capitals. So because they're showing the dominant, they have this dominant. And we can kind of backtrack and problem solve like that. So practice these Punnett squares. Practice these pedigree charts. I don't want to keep just talking and working through them, but use them like a practice problem. They don't just pop out right away. They take time to kind of problem solve. So spend some time and practice. So perfect. Here's a little bit of extra practice if you want, but that is it for chapter 2, talking about all this genetics and inheritance at the end of chapter 2. This takes a lot of practice. You saw those skills doing the Punnett squares or the pedigree charts. They're not overly difficult. However, it's making sense of all the terms to fit with it and understanding a little bit more in depth. And that's what makes it the hard part, is when they start to ask these skill questions or putting all these puzzle pieces together and how do they all work together, and thinking of how to tie it in with the earlier starts of all this DNA stuff to later, like we see this point mutation or frameshift mutation, but going back to these DNA strands and tying it all together. That's the difficult part with biology. So we should be getting ready for our quiz that will be coming up in June 4th and 5th. We'll have our chapter 2 quiz. We will be having our unit test on June 10th to 12th. And then our last day of classes are on June 17th, so we need to make sure all workbooklets are handed in that day. I'd prefer even on the 16th, have all workbooklets completed and handed in, and our marks or your final grade will be done on Thursday the 18th, and then that's it. Unless you're practicing or hoping to write the Science 30 diploma, you'll have nothing left after that. So take care and enjoy your summer.